Well, good morning, everyone. Welcome to day 2 of On Helix Digital 21. I'm Tony Jones. I'm Chief Exec of One Nucleus. It's my pleasure to be welcoming you this morning. We had a great day yesterday where we talked a lot, I guess, about the people factors and infrastructure and policy landscape we need to drive this sector forward and deliver better outcomes for patients. It was a great set of panel discussions that really set the scene for today, I think, where we switch gear a little bit and we focus much more on the technologies, the collaboration and the investment going into our sector that will enable us collectively to really make a difference to some of these global health challenges. And we've seen some great advances over the past year, of course. I think very timely yesterday, we saw illustration of where the life sciences in the UK sits in the minds of investors with the Mike Ward and Clarivate team with the BIA releasing their report, showing again this year for UK life sciences is heading for a record year of investment, showing the confidence and the value this sector can create. So I think we'll have a great day of sessions going through those different technology areas and funding areas. But I think without further from me, it's time for me to get things started and introduce our first speaker, an esteemed academic, fantastic track record, both in large industry, in startups and in academia, pushing back these frontiers of research and creating value, spin-out companies and ideas and innovation. So both in the UK and internationally, I think it's a great pleasure for me to introduce someone I consider a friend as well as a colleague for many years. And I'll just hand over to Professor Ian Charles, Director of the Quadram Institute. Ian.
Thank you, Tony. It's great to see you again. And thank you again for inviting me. It's a great pleasure to be here. I'll crack straight on with my presentation, please. So can I have the first slide up? I think this is a very relevant time to be looking at the whole life science sector. Is my first slide up? I actually can't see that. Have I got a view of the screen? And yes, here I see it. Thank you. That was just technically my incompetence. Apologies for that. Right, I'll crack straight on. Can I have the next slide, please? As I said, I've really got a strong background in this sector, and I really want to convey some of my enthusiasm for what's been happening. But this is a very personal context. It's been a very personal journey for me, looking at the way life science evolved over the last few years. Tony, thank you for the great introduction. Sometimes when people talk about me like that, I don't know it's me they're talking about, but thank you for that introduction. I have had a lot of time in life sciences. I've worked a lot in both translational basic research in big pharma and in spin-out companies. More recently, I've been involved in institute-level research. So a lot of what I'm going to be speaking about now is around institutes. I'm currently director of the Quadram Institute in Norwich. Many of you won't have been to Norwich, but I'll explain a bit about how the Norwich situation works. Before that, I was at the i3 Institute, and before that at the Wolfson Institute, where I first met Tony, Institute for Biomedical Research at UCL. I've been an entrepreneur, business leader, board executive for Arrow, Austerix, Longus, and been a non-executive director. I've got a lot of experience in the sector. Next slide, please. And I just want to give some of the context of that around what's been happening in life sciences and how we responded, particularly to the COVID-19 challenge, and how that spurred us on. There's other elements in this as well. And before COVID was the whole existential threat to UK research in the form of Brexit and how we were going to handle that. Next slide, please. So there've been a lot of updates and a lot of considerations of what's shaping our sector. And I love this Steve Bates quote from BIA around 2020 was really the year when biomedical innovation showed its true value. I think that's true. But before then, there's been a lot of thought and preparation around what's happening at Brexit what's happening in context now of the COVID-19. You're probably all familiar with what's been happening. There have been so many strategy reports. It's difficult to keep on top of everything in terms of the Life Science Competitive Indicator Report, the Life Science Industrial Strategy, that's the John Bell report, the Office and Science Technology Strategy paper, the Patrick Vallance report. Lots and lots of strategy documents out there, and it's really up to us to try and steer a way through this and actually what it means to us. Next slide, please. I think one of the things that neatly summarizes where we are with a lot of the work is actually around what's happening in the development of the life science sector. And what I really want to talk about here is the context of why it's a time to look to the future of life science research and what that actually means in terms of our interaction with other sector elements. So for me, that means looking at the landscape, MRC, BBSRC, EPSRC, all the great funding agencies that are coming together as part of the UKRI, the commercial sector, the charity sector. These need to interlink in some fashion. And I think we really need a better handle on how we're doing that. So on the one hand, we've got this great landscape. Next slide, please. And on the other hand, we need to steer that in with where we are with the life science reports, the role of government, the role of charities, how we see the individual strategies playing out in terms of the context, and the sense we need to make of it in terms of the research we need to do. And again, I think you're all familiar with what's been happening in the life science R&D roadmap. Many of you will have a much better handle on this than I have, but I think there's just so much going on here. This increase GDP 2.4% by 2027, 22 billion extra a year by 24-25. A lot of movement in the sector that we're going to need to harness and understand its potential. Next slide, please. One of the things that summarizes a lot of this is actually the Bell report showing these main areas: the science, the growth, the NHS, the skills, and this new area of data science and artificial intelligence that we really need to make the most of, and how we handle all of these new elements going forward. So again, many of you will have read that Life Science Industrial Strategy report. It's not the be-all and end-all of what's happening in life science. And I know that there are lots of elements that are not covered by this industrial strategy that actually seems a lot to do with the human health involvement. And that's understandable given the context of what's happened with COVID. But we need to stir into the pot the other agricultural areas, the other environmental areas. So there's a lot of bioscience that's not covered by so much by the industrial strategy in the Bell report, but it all needs to be stirred together into one big pot so that we actually get leverage from what we're doing. Next slide, please. And I think you can see that in this slide here that actually just highlights some of those elements. This is from the Grand Challenges document, BBSRC. I thought I'd just highlight this for you to look at, the Build Back Better concept in context of the BBSRC's bioscience strategy. It's very important for us to get into context these grand challenges and what they actually mean in context of all the other strategic elements. I happen to like this Build Back Better and the grand challenges because it does emphasize some of the bigger elements around this bioscience for sustainable agriculture and food, bioscience for renewable resources and clean growth, healthy aging, big concepts coming through here that we need, as I say, to stir into the pot when we're thinking about the strategy and the way ahead. I'm particularly keen on this angle, as you see, from my role as director of the Quadram Institute. I'll come on to some of this and explain what that means in context, because it does play directly to our strengths. Next slide, please. And actually what we mean by a life science direction. I'm again, I'll put some context around this now. Many of you will not have been to Norwich. Next slide, please. But I just want to show you with this, what a powerhouse of science is carried on at Norwich. One of the great things that people don't understand is there's not only a major hospital, there's a major university. There are 4 significant research institutes on site. And there's also, you can see in the bottom left-hand corner here, the Centrum, which is our bioscience innovation hub. So we've got all the elements here, I think, stirred into the same pot around consideration of hospital, university, institute research, but also harnessing this interdisciplinary nature of the innovation that we need to catalyze as a consequence of having this unique world-class cluster of science together. Now, I'm singing the praises of the Norwich Research Centre here, but I think this is one of the future directions of research. We know research is interdisciplinary now, much more so it was when I started my career in research. Next slide, please. And this is really exemplified by what we're doing on the Norwich Research Park. We're bringing together large clusters of research that have critical mass and can work together. Now, those clusters can work together because they can leverage the science, synergize the science from around each other. This slide just shows the new Quadram building. If you've not seen it, I'd encourage you to come and see it. It's an absolutely fantastic place. A new building. It's called Quadram from the 4 partners, the former Institute of Food Research, the BBSRC, part of UKRI, the hospital and the university, thus 4 partners, thus Quad. But it really brings together this whole idea of the impact of food on health. The great thing here is, I think we've learned from the, one of the lessons we've learned certainly from what's happened with the COVID situation, is that we're not a very healthy nation. One of the ways we can fix perhaps some of those health issues is by looking better at the relationship between food and health and the impact that has on the microbiome. And again, stop me from talking about the microbiome. It's one of my great loves going forward as a research area, but it will be something we want to look at with more detail going forward. So next slide, please. A little bit more about Quadram and the way it works together on the Norwich Research Park. We've got the largest endoscopy, one of the largest endoscopy units in Europe that handles around 40,000 patients. 100 clinicians, lots of endoscopy rooms, lots of labs, a clinical research facility, category 3 labs, ordinary labs, and collaborative and innovation lab space. So it really signifies to me that next direction, I think, of research, which is to synergize those interdisciplinary aspects of science. And we actually interact with the John Innes Centre, the Earlham Centre, the Sainsbury Centre, with the whole food health agenda, but we're also able to do clinical trials and able to leverage off some of those aspects of the clinical research interface. Next slide, please. And one of the ways I want to illustrate some of that work is how well we've interacted with each other, with the hospital, with the university, with the other institutes as part of our response to the COVID-19 situation. Next slide, please. Well, one of the great things we've been able to do is actually been able to respond, of course, very well as a consequence of what we're doing with the whole track and tracing of SARS-CoV-2. We were able to second researchers into the NHS labs. We were able to look at development of the whole idea of tracking and tracing genomic epidemiology. We've had members of WHO committees, Mark Palin, who suggested using Greek alphabet for the variations of concern, Simon Carding, who's been on expert panels, and Martin Warren and Simon again, who's been involved in vaccine design. Next slide, please. One of the great things, of course, we've been able to do is leverage off what we're doing with the hospital in terms of biorepository. Louise Jones is our director of the biorepository, but it's been able to get and distill some of the elements around of patient samples. And this includes getting viral genomes for us to sequence. Next slide, please. And this actually plays very well to the whole agenda of the Bell report and the Building Back Better concept of how we're synergizing research. We were able to work with Sharon Peacock's great UK consortium, the COG19 consortium. We've had experts on international panels looking at the way outbreaks have been appearing and disappearing throughout society. So this whole genome sequencing has something we've had in place anyway as a result of our own work through tracking and tracing. Next slide, please. We've also been able to support the NHS with this, of course, looking at outbreak control, both in hospitals, in factory sites, part of the REACT study. We've also, through a clinical trials unit, been involved in the Novavax COVID-19 vaccine, and volunteers have been through on vaccination trials. So again, great support, but great interface between research and the clinical situation. Next slide, please. And we're also doing a very interesting study, Arjan Narbad, looking at the COP study, looking at the way the viral genome can persist in fecal material and what that actually means, both in terms of the biology and infection, but also in terms of tracking and tracing in fecal samples. Next slide, please. So what's the way forward? Let's have a think about that now. So what do we really need as part of our next steps? I've described the situation on the NRP, the Norwich Research Park. I've described the Quadram. I think Quadram's a great model, synergizing with research across the board in the agri sector, the food sector, the pharmaceutical sector, the clinical sector. Next slide, please. Next slide. Yeah. Thank you. So what does that actually mean? Well, I think what we need is infrastructure. So we need people and talent, and we need those collaborations. So the next steps will involve critical mass, I think, is something that's often overlooked to drive interdisciplinary research. So we need the funding to drive that, and Quadram's a great example of that infrastructure. Next slide, please. And I like to place institutes at the center of that sort of model. If you look here, look at funding, look at the sectors, look at the resources, look at the challenges we need to meet. And at the center of this, I've got institutes. It doesn't need to be institutes in that context, but what it does need is to have facilities that drive an understanding of that critical mass in any situation. So I think institutes actually provide a good basis for the next steps in the development of the science we need. I think there are lots of really good reasons to do that. And we talk about now about technology sovereignty. We're talking about building back better. We're talking about a whole level of play that we've not had to do before. And that's post-Brexit and post-COVID. But we're really in a situation now where we do need critical mass, we do need skill sets to be exemplified, we do need a better interaction, not just between what we're doing with basic science and clinical science, but that basic science, food science, agricultural science, and informing on policy right across the board now. And we need centers to drive that interaction that have resource and skills that can synergize. Next slide, please. So I think the future of life science really needs to develop those strengths in places. We really need to empower not only that databases with NHS and the big data with NHS, but right across the board with genomic-driven approaches, with the agri sector, with the food sector. So this is really a fusion of ideas between those big pharmaceutical, food sectors, life science sectors that we really haven't brought together before. I think we need the people to drive that, and we need the places to drive that. So it's about skills, it's about collaborations, it's about understanding how we impact funding decisions, regulatory decisions. So again, what I'm arguing here, I think, is for a critical mass basis to actually take us to the next level for the development of science. Next slide, please. And just remains for me to thank you. I think I've timed that particularly well, Tony. So thank you, everyone, for listening.
Ian, thank you. Yes, as you say, perfect timing, right in the slot, and give some time for questions which have come into the chat box. I guess one that was on my mind all the way through when you were looking at the response to COVID and what we need in the future in terms of different disciplines and critical mass. I mean, we've seen, I guess, what we would call open innovation at a scale we could never have imagined for international biopharma and medtech. When you get to that critical mass, and now you've built a community in Norwich, if you like, of critical mass at that level, what do you think the impact of the pandemic, but also Brexit in terms of our international connectivity and being part of that critical, what does that mean for UK life science?
It's unfortunate that there have been cuts in the ODA budget, as I'm sure has been discussed widely. But we need to absolutely maintain those critical connections with national and international partners. You can't see any way back from that. But increasingly, I hear an argument of national of some form of sovereignty around the science that we need, which is certainly true. And I think COVID has taught us we need to be generating new ideas, but also have the industrial basis to manufacture those bases. That's certainly true. It's a much more complex argument. If you end up spending more money at home, inevitably, you're squeezed and you don't have as much money to maintain those international collaborations. I think this is something that probably needs wider debate, Tony, in order to thrash it through to understand what proportion should be spent where.
I think it's a balance we're going to have to work out, because as much as that phrase of no man is an island, I guess no country is an island.
No country is an island, yet we do need this technology sovereignty in some way. And I think that is one of the lessons we may well have learned from COVID. We need to be able to generate the ideas, hypotheses, test them, and then generate them, produce them on an industrial scale.
Plenty of challenges ahead, but I'm sure with institutes like Quadram and many others as those hubs, which I tend to agree with you, we need, then I think we've got a great chance of addressing some of those big challenges where we don't just need genomics, we need the data science and we need manufacturing. Absolutely. So I think that sets the tone really well, Ian. Thank you very much for taking the time to do it. I'm sure there will be more questions people would love to ask, but as always, there's never more time. And
thanks, Tony.
Waits for nobody, so we shall bid you farewell. Thank you.
Bye.
Which moves us on to, I think, one of the real advances we've seen over the past 12 months was mRNA. We had many terms that many people weren't familiar with. Furlough came from nowhere and we didn't know what it meant. In the science space, mRNA, I think it was Kelly Shervick in Science in December last year said mRNA had tantalized pharma long before the pandemic of its potential applications in vaccines and in drugs more generally. So now I'm delighted we have a drug hunter of vast experience and now leading the European external innovation sourcing for Lilly in Tim Luca to lead the next panel discussion through mRNA beyond vaccines. Tim, over to you.
Yeah, thank you, Tony, for the great intro, and obviously thanks to you and the whole One Nucleus team for inviting me and putting this panel together. Yeah, I'm a VP in Corporate Business Development and Venture Science at Eli Lilly. We've invested substantially in oligonucleotide drug discovery and drug development over the past few years, many announced deals and partnerships. I guess, I'm just reflecting that, these are large, unstable molecules. Research started in the 1960s, and these days, obviously, we hear a lot about the successful COVID-19 vaccines based on mRNA technology, but there's so much more to this field. And so I'm really pleased that we've put together such an illustrious panel, many joining from unsociable time zones. So thank you to all of the panelists who've carved out some time to discuss this with me. So, without further ado, I'll just run through who we've got on the call, and then I'll ask each of the panelists to introduce themselves. So we've got Stephen Powell, who's CEO of eTheRNA. We've got Klaas Zuideveld from Versameb, Richard Wooster from Translate Bio, Tadas Trunca from Thermo and Dimitrios Goundis from Exacis Biotherapeutics. So perhaps I could hand over to you, Stephen, to kick us off and introduce yourself.
Yes, good morning and good afternoon to everybody. And it's a pleasure to be here on the panel today. My name is Stephen Powell. I've been the CEO of eTheRNA Immunotherapies based in Belgium for the last 2.5 years. eTheRNA Immunotherapies is an mRNA company. We've been in existence for 7 years now. We have 3 technology pillars or interests. mRNA chemistry, of course, but also formulation and also process technologies and manufacturing. And these support a 3 development platforms, 2 in oncology, which comprise therapeutic vaccines and oncolytic work. And the 3rd area is in prophylactic vaccines in infectious disease. Thanks very much.
Thank you, Stephen. Perhaps Richard, I could get you to go next.
Good morning, good afternoon, everyone. It's a real pleasure to be here. Thank you, Tim. Thank you to the organizers for the invitation today. And I'm Richard Wooster. I'm Chief Scientific Officer at Translate Bio. And we are another mRNA platform, with our platform initially being incubated within Shire Therapeutics, actually starting in 2008. And from the earliest days, thinking not only about what therapeutics, what diseases to target, but also, as Steve mentioned, the manufacturing component. How can we manufacture molecules, the delivery vehicles, such that we could actually move them into clinical studies? We acquired the platform from Shire in 2017, and since then have been developing a number of strands of our work. A major area of focus for us are therapeutics for pulmonary diseases, where we're mostly using an inhaled route of delivery. And our lead program is in a phase 1/2 study in cystic fibrosis, where we're really encouraged by the safety tolerability profile we've been seeing, which is the design of this study, from not only a single inhaled dose of mRNA, but actually multiple doses, which have been inhaled by patients. And we believe this is the first time this has been demonstrated. And beyond cystic fibrosis, we have work in primary ciliary dyskinesia, another genetic disease, as well as other pulmonary settings. The second area, building into a number of different components, are liver diseases and how we're tackling those. And I think what I'll wrap up with is we have an active collaboration with Sanofi Pasteur on vaccines, where we have a COVID vaccine in clinical studies. And last month, we announced the start of our flu vaccine clinical program. So really an exciting time for mRNA. I think having now seen the success coming from the COVID vaccines, from Moderna, from Pfizer, I think setting the stage now as to how we can apply this technology in many other settings.
Great, thank you, Richard. Perhaps over to you, Klaas.
No, thanks, Tim, and it's a pleasure to be on the panel. So my name is Klaas Zuideveld. I'm the CEO at Versionmap. Versionmap is a next generation RNA therapeutics company, and our 2 lead assets are in uro-oncology as well as in urogynecology as well as in oncology. Versionmap has a proprietary platform to modify the sequence code of sort of the flanking regions of the mRNA, really to get the cells to do something different than it would normally do. So we can, for example, enhance the expression or the secretion, as well as target multiple proteins at the same time using a single construct and reduce immunogenicity. In our lead indication, structure and incontinence, we've recently shown we can actually regenerate the urinary sphincter reducing the involuntary loss of urine. And in oncology, we can actually target or shut down an entire pathway by targeting 4 proteins in one go. So we're very excited. I think both programs we're pushing forward into preclinical talks and hopefully next year, phase 1 studies. Thanks, Tim.
Thank you, Klaas. Tadas?
Thanks, Tim. Happy to be on the panel. Hello, everyone. My name is Tadas Trunca. I'm a senior manager for product management organization within Nucleic Acid Therapeutics business unit at Thermo Fisher Scientific. So Thermo Fisher Scientific is a company quite well known in this field, in this industry. We're actually, the world leaders in serving science. And we do that through multiple different business segments and units and etc. But at the end of the day, we're actually we're leading the industry through the scale of our capabilities and breadth of our capabilities. And what do we mean by that? In particular, we've been, innovating as well as growing with the mRNA field for many years. The mRNA technology as such has been, there for more than 40 years, right? So we were, partnering and innovating together with the field in supplying different components and solutions and tools for the market. But really, with the recent company growth, we have quite significantly expanded our footprint. Our newly formed pharma services group, a former Patheon, which you may have heard of as well, is helping mRNA therapeutics and vaccines pivot, right? Scale in the market and etc. Thermo Fisher, through the Life Technologies acquisitions and a couple other organic businesses growth, we've actually been working with the companies in the field from a very, very early, I would say, research through clinical and preclinical studies and pivoted to there. So Nucleic Acid Therapeutics business in particular, our expertise and our capabilities and capacities really reside within supplying those key raw materials for mRNA drug substance production. But as you think of the Thermo Fisher Corporation, in general, it's way more than that. Through the CDMO services from Pharma Services Group, we offer a lot of different solutions to customers. So happy to be on the panel. Back to you, Tim.
Thanks, Tadas. Last but not least, Dimitri, I'm not sure if you're going to share a few slides or whether you're going to speak, but over to you.
Yeah, maybe I'll say a few words first, and then I can go through some of the slides. So very nice to be on the panel. Thanks for the invitation. My name is Dimitrios Goundis. I'm the Chief Business Officer and Head of Finance for Exacis. Exacis is a company that was founded last year in 2020, based purely on mRNA. And the technology came from another company that was based in Boston, in the Cambridge, Massachusetts, called Factor Biosciences. And what we've done is we have licensed the technology, which allows us to use the mRNA to produce iPSCs, which are induced pluripotent stem cells, based from somatic cells. So we do all the gene manipulation with the mRNA. And moreover, we also have developed an enzyme that allows gene editing and cell engineering that, again, is all based on the mRNA. The advantage of that being the fact that it's completely DNA and footprint free. We apply this technology to produce iPSC-derived NK cells and T cells, and obviously to do either NK cells with CARs and T cells with CARs, or just pure NK cells, which would then be used with monoclonal antibodies. And we target tumor indications, both solid as well as hematologic tumors. Now, I do have a little presentation. I'm not sure if this is the time to do that now, but happy to go through the slides if that's, the slides basically provide a bit more information on our technology and a bit of background on the company.
Maybe if there's one slide, Dimitri, you wanted to call out, and then we can obviously bring out this in the discussion.
We got to slide 6, actually, which basically is the whole summary of the 4 elements of the technology. If I can have the slide 6, please. I think it's 6 anyway. Yeah, that's the one. 1 second, let me pin on it. Okay, so yeah, as I was saying, so there are 4 elements in our technology. One is the mRNA cell reprogramming, and again, that basically is used to attack proteins such as OCT4, SOX2, KLF4, CMYC, and Lin28. Then riboslice is again the mRNA vector that we use to target the genome. Novoslice is actually the gene editing protein, which again is based on mRNA. All these are synthetically produced by us. And then we use what we call a tornado system. Again, if you just look at the name tornado, RNA is included in that. And that's a proprietary system that we use to deliver the mRNA to the cells. And the advantage of that is that it's not inhibited by serum, and it's a very efficient way to deliver the mRNA. I think that's basically the essence of our technology. And as I said, we apply this to producing iPSC NK cells and T cells, and obviously we target tumors, both solid as well as hematologic. But all the editing, all the manipulation of the cells is done by mRNA. So that was it. Thank you.
Thank you, Dimitri. And just a note for the audience, we have a chat function. So if you have any burning questions for the panelists, please send them in, and we'll do our best to incorporate them into the discussion. Great. So I thought, we've obviously discussed behind the scenes a few questions, but I thought we'd start gently from the basics, because there's many people here who perhaps haven't worked with mRNA. Myself, I've never touched an mRNA molecule in the lab, and it's only relatively recently, through a lot of reading, I've come up to speed. So perhaps I could get maybe Richard and then Stephen to just talk through the evolution of mRNA as a therapeutic modality, and what we can do with it, what kinds of biology we can open up, and then obviously we can start getting into more and more detail as the panel goes on. So perhaps I could start with you, Richard. I know you've got an illustrious career in this space, so perhaps you could give us your take on 1960s to the current day.
Wow. Yeah, so Tim, I did have that fortune of being able to work with mRNA in the lab a few decades ago now. And certainly what I recall from that time is mRNA is an incredibly labile molecule. You almost just had to look at it and it would start to degrade. And also the really small quantities that we would be working with. And so I think to me, two big evolutions of this technology. One is that ability to, through in vitro transcription, manufacture the mRNA to a quality that you would want to use in clinical studies. But the other big component is then delivering the mRNA to the cells to actually transport it into the cytoplasm so that it can be translated into the proteins that you want. And that is certainly a big area of focus for us on the chemistry of these proprietary and novel lipids, which we incorporate into our lipid nanoparticles. Because what we need, and I'm going to focus here on our pulmonary setting, if you think about the journey that mRNA is making, so it's being created in the lab, it's being packaged into the LNPs, we then nebulize those LNPs, and we're using a vibrating mesh nebulizer to do that, so to create this fine mist. And there are shear forces in that. The LNPs and RNA have to survive through that. Then, as they move down into the lung, of course, in our lungs, we have a mucus barrier, which is there to protect us from bacteria, from dust, viruses. And so the LNPs have to penetrate through that. And so we worked around how to penetrate through that layer and actually be taken up into cells. And not just thinking about in healthy lungs, but when we think about cystic fibrosis, I think we know that one of the key sort of aspects in the lungs of patients is that the mucus is far thicker. It's actually a more challenging barrier for us to work through. But having penetrated through the mucus, then seeing the translation in cells, and it's not just any cells, we're now getting into, well, how many cells are we actually achieving expression in? What type of cells? Whether it's the ciliated cells, whether it's the AT2 cells, for example, in the lung, so the location where you want to achieve expression. And then just one final thought on the delivery vehicle. it's great to get the mRNA there with the delivery vehicle, but then we want that delivery vehicle to degrade, essentially to be cleared from the body, because it's served its purpose. And so what we have in our lipids is actually now engineering points of metabolic liability. And in some ways, this is counter to many of these sort of small molecule drug discovery approaches where you want to maintain your molecule. We're actually looking for a rate of degradation, ideally, which clears the lipid before we have to redose the patient. So we avoid accumulation over time. And so it's now getting to a stage where I think we can move from the single or the two-dose applications that we've seen in the vaccine space into a chronic setting of mRNA therapeutics. And I think this is now the exciting stage where we can design and then start testing and taking forward these products, which patients might take for the rest of their lives, and having that safety as well as the clinical benefit that comes from those types of products. Tim, back to you.
Yeah, thanks. Maybe Stephen, obviously from the most simple level, you put the message into the cell, you restore protein levels. I know there's an awful lot more to this. I think you probably only have to look at a mRNA molecule, which is sort of made to order in cells and then falls apart after it's been used. I guess there's a lot more to getting into drug discovery in this space.
Yeah, absolutely. And I think as Richard has said, there's an underlying requirement for a toolbox with three main components, which is the mRNA chemistry itself. How do you optimize that expression, and what are you expressing? How do you formulate it, as Richard has just articulated, and of course how you manufacture it, which I'm sure Tadas will have something to say about that particular area as well. We have now a significant momentum behind mRNA, not just in prophylactic vaccines, but also in other immunostimulatory routes. In eTheRNA, we're using mRNA as an adjuvant to additional mRNA. So we're looking to amplify dendritic cell activation and T cell proliferation by using a very specific combination of mRNAs co-presented with antigen coding mRNA. And I think one of the things that gets forgotten quite a lot is one of the key advantages of mRNA technology is you can actually code for antigens which would be otherwise quite difficult to manufacture. So in the case of certain peptide-based antigens, very complex peptides can be very difficult to manufacture from FMOC chemistry as an example. But you can code them into mRNA and then get a good level of expression from that. I think what is increasingly exciting now is how mRNA is being used in a number of different areas, not just in the vaccine area, whether it be therapeutic or prophylactic, not just in protein replacement, but actually starting to see mRNA now being applied to other therapeutic areas. We heard from one of the other panelists talk about urogynecology applications, and I suspect we will see that application for mRNA technology broadening out. And whatever happens as to how we extend this modality, we are going to continue to need to expand and invest in the formulation approaches and the delivery approaches. Can we make mRNA more targeted by making its formulation more targeted? The answer is almost certainly yes. Can we do smart things with the mRNA itself in terms of circular mRNA or self-amplifying RNA? The answer is almost certainly yes. And I think the next decade will see a significant expansion in the technology base, but we will still have to master some of those basic elements, particularly the formulation and the manufacturing, and perhaps come to the conclusion that one size does not fit all, The formulation needs to be adapted for very specific routes of administration, and adapted for very specific biological settings. So I think there's a huge amount of work to be done. What has been shown from the last 18 months is that mRNA is a fundamentally safe modality, but then how do we optimize its efficacy? And I think that's the key challenge for this next phase of the technology.
Thanks, Stephen. Yeah, that brings us nicely on, I think, to we'll come to you, Tadas, for the downstream problems in a minute. But Klaas and Dimitri, I mean, Klaas, you're working on, I heard, combining different mechanisms into perhaps one molecule, and also delivering to sort of the Euro system. I guess in some applications you need highly immunogenic, foreign mRNAs. In others, you need to be very safe. And I think, Dimitri, you're working on using these to form allogenic CAR-T and CAR-NK cells as the modality. So perhaps Klaas and then Dimitri, you could chat through some of the science that you're doing and how you're leveraging mRNA to achieve that.
Mm-hmm. Oh, thank you, Tim. No, I think that's exactly it, and I think also maybe tagging on to what Stephen said. I think obviously in the last year, I think over billions of people have actually received mRNA into their upper or into their arm, into their muscle. So I think it's also having. I mean, it's not. I think it's part of it's the deep expertise of sort of understanding the physiology, particularly what we're applying in sort of urogynecology. It's having an understanding of how muscle tissue works, being able to sort of tag into those systems, and then at the same time sort of modifying the mRNA to sort of increase its potency. We only need to get very small amounts of mRNA into the cell for actually to get its function to be able to regenerate the sphincter back, which is kind of a unique technology, which is something you wouldn't be able to do with say a protein by itself or a small molecule. So I think those are sort of terribly exciting fields, and I think it tags on to the right sort of variety of things or directions we can go into sort of with mRNA. Maybe speaking to, I think what you mentioned, I mean, obviously one of the other things we're doing in oncology is obviously a very crowded space. That's where we said, well, how can we do something sort of transformational? And really, I think also by using some of that expertise or the understanding of the biology we have, is how can we just combine different targets in a single construct? And for us, having it in a single construct also ensures that in a very sort of heterogeneous space, you can actually get all the targets in, or target all your molecules at the same time. So if you want to shut down an entire pathway or give the tumor no other options to, for example, get around to circumventing the pathway you're blocking, is really to see if can we block everything at once. And so by going into with a single construct and doing that at once, we believe is going to be transformational in this space. But it sort of tags on to, I think, a lot of the innovation is going to be in sort of some of these newer technologies, whether it's modifying these flanking regions or trying to do something else with the mRNA. I think that will get us there. And I think having a deep understanding of biology is key, both for the tissue as well as what you're trying to do with the mRNA. Thanks, Tim.
Thanks. Dimitri, do you want to say a little bit about what you guys are doing?
Yeah, so, we are dealing with very tiny amounts, right? Because what we're trying to do is we're trying to select the right clone that has the right edits. And what we find is that the efficiency of the mRNA is pretty good. We have, we can do up to 5 repeated transfections, and we have more than 10% efficiency. And then what we do is we select the clone that has the right edits, and then we expand that. So we're dealing with very, very small amounts, obviously. Our mRNA is also synthetically produced, and it's produced in a way that minimizes its immunogenicity. And the advantage of using it in our setting is the fact that any editing that is being done to the cells is completely footprint free, right? Typically people have been using in the past, viruses to deliver loads to the cells. And there is always the risk there that as the DNA gets into the host cells, you might end up with certain mutations, and after repeated cycles of cell growth, those may become problematic. With our setting, with the mRNA being transient, doing the edit, getting the cell, getting a single cell, and then growing it, we know for a fact that it's devoid of any off-target effects, and basically we have a very clean product, if you like, which I think is really important for the indication that we're targeting. And also it gives us the flexibility to do several edits at the same time, and also insert more than one cut if we want to. So it's a great way to engineer the cells in our hands.
Thank you. And Tadas, I guess you get to hear about all these very interesting discovery projects eliciting maybe transformational biology, but sooner or later someone has to make this stuff and set up the supply chains and work out how to manufacture this on a suitable scale, perhaps for clinical trials, perhaps under GMP. Perhaps you could give the audience a sort of flavor of what you think is needed in this space and what it takes to be successful and what you guys do.
Sure, Tim. So I think this is a very, very broad topic, Tim. First of all, right, what we've seen for many years in mRNA therapeutics and vaccine development field, most of the manufacturing scales were relatively small, right? You did, which was basically feeding and seeding preclinical research and preclinical studies in general, and very early stages clinical phase 1 or phase 2. So really, up until like 2020, it was still getting the momentum. The manufacturing setups from a scale perspective was, as I mentioned, relatively small. I would say, microliters to milliliter scale. But as the emergence of COVID pandemic definitely did give a lot of momentum to set up infrastructure and supply chain as a whole to produce vaccines, to treat pandemic in general. So we as a Thermo Fisher, being partner to many companies and in supplying services and components, we've seen that evolution, right? The evolution that was expected to last several years as a minimum, to be squeezed into a couple of months. What does that mean? Simply, mRNA as a technology has never been before tested in a large scale setup, infrastructure and network, right? And a lot of agility for companies like Thermo Fisher and biopharma companies developing the drugs and the vaccines was needed. From a manufacturing perspective, the flow of the producing mRNA is relatively simple and well known and documented. You start from your plasmid production by defining your strain, which could be E. coli, for example, as a base. You then do you insert your gene of interest there, of course, in the plasmids. You do your cell-free mRNA synthesis in IVT vessels and single-use potential vessels. You purify that mRNA. You get rid of all the residuals or impurities there, and you use that to formulate in your LNPs, right? So definitely, I would say what are the key differences and needs when you scale from a microliter to tens of liters scale in production. From a supply perspective, you need to have agility to keep up, right, with the needs. Supplying way more enzymes, supplying way more nucleotides, which are building blocks of mRNA. This is where the company or where the industry in general pivoted to bring new breakthrough innovations in manufacturing floors. For example, Thermo Fisher, we did use our proprietary single-use manufacturing systems to produce large industrial scales of enzymes in GMP quality to be suitable for drug substance manufacturing. And we do apply also a large-scale GMP production of nucleotides, right? Just to support that drug substance production. So this is very, very important. The other thing, Tim, is from outside of innovating internal manufacturing processes to really yield large, pure products, large volumes of those, is the needs from a pharmaceutical industry. And those needs are typically related with security of supply, right? Thermo Fisher is uniquely positioned there simply because we have a wide global infrastructural systems in place. We do have our manufacturing sites based in different geographies in Europe, in the US, where we produce not only components for drug substance, not only plasmid DNAs, enzymes, and nucleotides, and modified nucleotides, or LNPs, for example, but we also offer drug substance or drug product production services. So from that perspective, it's really, really well coordinated, and pharma is really valuing the security of supply when it comes to picking which continent, which region, which manufacturing site, which country you want to buy from. So this is something that is really universally needed, I would say, from a manufacturing perspective, to have those options, to have that flexibility and assurance of supply, and really mitigating any sort of supply chain risks of not having the product on the shelf. This is something that any company should avoid indeed.
Thank you, Tadas. And I guess in common with any drug modality, I mean this is a very expensive, on paper at least, a very expensive type of therapy that's quite complex. And obviously, to open it up to the masses, one way is obviously to choose the right indications for the biology. But I guess another way is just back to basics, that to lower the dose as much as possible is going to help people like Tadas to make these RNAs. And I guess what I was hearing earlier, well, there are probably three ways to sort of combine to do that. There's obviously boosting the efficacy, the ability to translate into protein. There's optimizing the chemical and metabolic stability, and then there's obviously optimizing efficient delivery of the molecule to where you want it. So perhaps I'll get some of the other panelists to reflect on how they go about pulling that kind of story together. So maybe Klaas, to start with, I know you've been working on enhancing the ability to translate into protein in versameb. So perhaps you could say a few words about how you go about that and what's on your mind in that space.
I think so. I think that's really important, because there's two things, right? Is that you also don't want to get too much mRNA into the cell, because you're not a virus, right? You don't want to hijack the cell's sort of protein production mechanism really to shut it down to actually only produce your one protein. You want to maintain sort of the physiology. So I think that was also the leading thought behind us trying to say, okay, how can we do to actually get as little as mRNA as possible into the cell, but still be sort of very effective sort of clinically or therapeutically. And that's why sort of the whole thinking behind actually enhancing the expression, enhancing the secretion, it actually modifying the sequence code so that the cell can actually do that, with the end result of sort of creating a higher bioavailability of the, in this case, of secreted protein outside of the cell. You're actually increasing the potency of the protein tremendously. By doing that, you obviously need a much lower dose, so that further reduces the cost. I mean, the cost, I would say, the cost of goods for mRNA, I think they've recently sort of gone up so in the production process, but it's not prohibitive as such. So I think you can still produce very effective, therapeutic doses at a very reasonable sort of cost. I think the real cost probably sits somewhere else. So I'm not too concerned about this. So I think it's definitely feasible to actually have very effective therapeutics at a very reasonable sort of cost of goods. And I think the other thing is really to be creative, right? I think it's a lot of it actually sits. A lot of the problems are sort of purely biological. Having a good understanding of that biology will help you. Will actually choosing your indications wisely. I think it was mentioned before. Will get you there.
Thanks. Maybe Stephen, do you want to reflect on the sort of efficacy, stability, target engagement story? I guess the better and better we can get at this, the lower the doses and perhaps the safer the therapies are. So perhaps you want to talk through that. You might be on mute, actually.
Sorry, as the other panelists have commented, a lot can be achieved by optimizing the chemistry of the mRNA backbone to maximize the expression. But equally, formulation is, as we've all recognized, is something that is very, very important, not just in terms of the stability of the mRNA itself, giving it the opportunity to do its job, but also if you get the formulation wrong, you can impact on expression as well. And so those two features of the mRNA chemistry and the formulation really need to go hand in hand. And I think coming back to one of the things I said earlier is What we have found over the course of the last 3 to 4 years is that the route of administration, the indication, the application all have very significant impacts on the choice and optimization of the formulation itself. If you get that wrong, then the expression isn't strong enough, isn't good enough. So you either end up putting too much mRNA in, which we're all agreed with nobody wants to do, or you just can't get the right levels of, in our case, immune response. So it's a very, very fine balance. And I think collectively we're getting better at it. And I think the last 2 years has certainly enabled the technology to catapult forwards quite dramatically.
Great. I mean, Richard, I know you guys have been pioneers in sort of delivery vehicles, lipid nanoparticles for many years. Again, perhaps for audience members who really don't know anything about this space, could you just chat through, what these things are, how they work, how difficult is it? I guess many of these smaller particles naturally end up in the liver, and perhaps we want to avoid that in some cases. Others can be toxic. Others need very specific routes of administration and distribution profiles. But I must admit, I wasn't aware of it till I got into this space. I hadn't realized the literally thousands of publications and disclosures in this space. Perhaps you could give us a sort of flavor of what it takes to come up with suitable delivery vehicles in this space.
And maybe if I could, Tim, but before getting into that, I just wanted to, I think, add on top of what others have been saying on the mRNA component and how you can increase the potential benefit. So two thoughts there. One is the codons that we actually use within the mRNA sequence. And so we're not using the wild type mRNA sequence, but we're changing the codon usage, still coding for the wild type protein. But through that optimization, we can increase protein expression from each molecule of mRNA. And so that coming to how you can reduce the dose. The second aspect, and this comes into the ability now to make tens, if not hundreds, of different mRNAs for the same program. Almost, if you like, getting into the lead optimization that you might think of with a small molecule. And so we've been looking at subtle but important protein engineering components within that final product. And so, for example, to get the duration of expression, you could think of editing a lysine, which is known to be ubiquitinated in a protein, and so slow down the rate of degradation of the protein you're producing. And in that way, extend the duration of activity, but also that could lead to reduction in dose level as well. So I think that the way that the technology is evolving to now getting into these more higher throughput discovery experiments is enabling that. But to the LNPs themselves, absolutely, I would say there are many, many thousands of publications, and people have explored polymers, for example, in formulations, as well as a whole range of novel lipid constructs. And those come with their sort of advantages, but also disadvantages. I know you asked about liver. I think it's one, I remember from a number of years ago talking to people at Alnylam, saying if you can figure out how to deliver anywhere else than the liver, you could have such success, because the liver does what it is designed to do, sort of evolutionary, will take up nanoparticles. And certainly we're leveraging that within our liver programs. I think opportunities, maybe in a sort of slightly tangential way to how to achieve selectivity, I'm going to come back to the RNA sequence. And that is designing the mRNA to limit its translation in certain cell types. So putting in microRNA binding sites, other regulatory elements. And so it's what we have is a combination product which allows us to work in different ways to achieve that specific, whether it's organ or cell type expression. Because what we've seen is we've screened through many hundreds of novel lipids. what we find is that some work exceptionally well in some cells, in some particular organs, but that doesn't translate into other settings. So for example, we know a number of our very exciting pulmonary lipids actually are not good at delivering to the liver if you give them intravenously. And so we're really starting, I think, to understand now some of the SAR components of the lipids and what drives the benefit in different positions. And I think picking up on a comment from Steve, formulation is then just a whole nother layer that you can bring into this. And for example, we've seen where we can take the same ionizable lipid in our lipid nanoparticle, change the formulation, and see 2-3 orders of magnitude of increase in protein expression just through the subtleties of the formulation. And so I think this opportunity to scale, because when we think about vaccines, they're usually given in the maybe tens of micrograms of mRNA. For therapeutics, it's often you might think of perhaps milligrams, even tens of milligrams. And so the scale becomes very different. So addressing all of these components, how can you reduce the scale, enhance delivery, is going to bring benefit in a very wide sense.
Thanks, Richard. We'll come back to this, I think, with a few of the other panelists. But Dimitri, you're going to circumvent some of these issues by delivering a cell therapy. Perhaps you want to talk a little bit about that.
Yes, yes, thanks, Tim. Yeah, well, in our case, we are only dealing with tiny amounts, right? And so we don't have to worry about delivering the RNA or the mRNA to an organ and then worrying as to, you know, the amount and what's the per cell delivery to that particular organ. You know, it's a much more complicated setup. In our case, we're dealing with single cells. And so in a way that makes it less of an issue. So our key thing is to make sure that you know, when we try to transfect, let's say, you know, 100 cells with our product, that we have as good an efficacy as possible with our LNP, and that we've been able to achieve. You know, and that basically allows us, as I said, you know, to take that particular cell that has been transfected to expand it and use it. And at the same time, when we deliver a CAR into a cell or a cell edit, again, you know, the mRNA that's being used is delivered in the same way. We still have the same efficiency. And actually, when we're dealing with a single edit, you know, the efficiency is over 60%. So basically, we can take, you know, 100 cells, and we try to do, you know, let's say that we would like to, you know, we want to silence one of the MHC genes. You know, we deliver, you know, with the mRNA, the actual sequence that will result in silencing of that particular gene, and then the efficacy goes up to almost 80%. So it's a very efficient way. You know, the way we use it, basically, the way we use mRNA and the delivery system, it's a very efficient and effective way to produce the edited cells, and not only, you know, in terms of editing in terms of you know, silencing certain genes, but also editing in terms of expressing certain genes. In our case, the CARs.
Great, and I guess Klaas and Steve, strategically, I mean, there's been a lot of deal making in, you know, delivery vehicles. You know, LNP deals are getting increasingly expensive, both platforms being acquired and, you know, perhaps licensing individual LNPs that have been optimized. But for your companies, is the strategy to sort of partner a bit later when you've got, you know, your clinical candidates coming on stream, or is it to develop proprietary solutions in that space yourselves? I guess that's a key question for, you know, emerging biotechs in the mRNA space. So I just wondered if you could chat us through what you're thinking and you know, some reflections on that.
Thanks, Tim. Do you want me to go first?
Yeah, please.
All right, thanks. So I think, I mean, obviously, I mean, we have I mean, also Versionmap has actually developed a proprietary platform. I think by now we've actually developed, a huge number of molecules in-house. All the IP is developed in-house. So I think at this stage, we're really sort of seeking financing really to finance our clinical program in stress urinary incontinence. I mean, obviously, we're open to the partnering discussions, whether it's in some of our other sort of indications or oncology. I think in oncology, obviously being able to combine different targets, opens up the space to actually generate a huge number of molecules. So we can impossibly develop everything ourselves. So I think we're very much open to that. I mean, we do. I mean, without wanting to say too much, we do get approached by pharmaceutical companies are in active discussions with them. In parallel, though, I think a large part of the strategy is also to advance the company further and really to get to clinical proof of concept, really with the objective to actually deliver a therapeutic which can help. I mean, in the case of stress urinary incontinence, help millions of women worldwide, and in the case of oncology, really to deliver something transformational.
Yeah, and I was thinking more, sorry, I may have misspoke on the question, Klaas, more about your strategy for bringing in a LNP type solution to your company. Would you develop that yourself, or would you, discuss with others? Because obviously you need both parts to play their role in coming up with a therapeutic. That's more what I meant. Sorry.
So, okay, sorry. And also specifically on the LNPs, I'll say this, for our 2 lead indications, we're actually not using LNPs. So the modifications we've made to the mRNA are actually purely sort of biological, and having those biological modifications actually allows us to get around having to use a formulation per se, which I think has numerous advantages. I mean, obviously, I think one of the, and I think this, and it sort of comes back to I think what was mentioned earlier in the conversation. If you're thinking of sort of targeting specific organs, looking at sort of systemic administration. I think that's it's an area probably which needs to develop, and that's where actually look. I mean, that's I think where we'll probably look for work with a partner which can very specifically target a particular organ. But in all honesty, I actually haven't seen. I mean, I've seen one thing which is impressive, but I haven't seen very much. So I think that's still. I mean, that's still a number of years out. So I think having an understanding of the organ or the biology you're trying to target and actually trying to find a solution for that rather than trying to come at it with a formulation approach, I think that's been our strategy.
Great. And Steve, any thoughts on strategies for delivery solutions for you guys?
Yeah, so we originally set off to partner and bring in LNP technologies rather than do it ourselves, but we ended up doing it ourselves. So that we could achieve the very specific levels of expression and performance properties that we were looking for in immunotherapy. As a result, using indeed the sort of SAR type approach, we've actually built up good libraries of LNPs, and so we can certainly utilize those ourselves. However, what we have found is we've actually done quite a lot of partnering and licensing around lipid components, coming to one of the points that Richard made, so that we can get the right level of degradation profile in those lipids. So we've actually brought in very specific lipids, in some cases novel lipids, to impart very specific properties onto the LNP. We're also partnering with a number of institutions and partners in terms of looking at next generation formulation, which may not be LNP. It could be different forms of polyplexes, as an example, and how we can decorate and target those. So partnering for us, because we're such a small company, is so key. But we've been able to use it, I think, to everyone's benefit. And I think there are so many technologies out there that partnering is almost inevitable.
Tadas, this all sounds pretty straightforward. Just mix the mRNA with the delivery vehicle and off you go. I guess there's a bit more to it to get this straightened out to to GMP quality, etc. I guess you need things to remain at least stable, reasonable shelf life, those kinds of things. How does all that work?
Sure. Let me maybe start in from the core question. What's beyond the COVID-19 vaccine, right? What's next? So mRNA as a new pharmaceutical pillar, right? actually has a lot of potential to be used in multiple different infectious disease areas, right? Protein replacement areas, allergy, tolerization field, gene editing, gene engineering. So a lot of, from a field of use perspective, a lot of different areas or diseases to treat. And the reason, Tim, why I sort of start from here is when you think of that, you have very different addressable patient populations, right? And with that, there is potentially some opportunities how the manufacturing as well as supply chain systems will be set up. For example, if you're company working towards individualized cancer vaccines, for example, you may need a very, very different set of infrastructure to build that unique set of mRNA for your patient, versus where you have a global widespread infectious disease where you need very large infrastructural designs and setups. So, from a manufacturing perspective and a scale-up perspective, I think the industry will see a couple of solutions to be implemented. Automation, for example, is definitely in the radar for many companies in the field, how to automate the process, how to make it more automated, less risky to any external factors that could affect your drug product and drug substance quality to some extent, or purity. So automation could definitely be a very interesting field for those vaccines or therapies that could be more or less individualized, for example. When you think of a large-scale production of vaccines and therapeutics, definitely, typically it's being segmented in a couple of segments. The way, early stages of the process, plasmid linearizations to mRNA synthesis and capping of that mRNA molecule is definitely within the GMP requirements. Even when you think of raw materials, for example, we are cautious of where those components are being used. Purity of the actual mRNA, the synthesized mRNA, but also components that go into building that mRNA is critical. We do not want to see any impurity profiles when you have the final drug substance built that you have to purify and spend additionally in purifying that through HPLC or other systems. So something bear in mind as you think, as Klaas mentioned, cost of goods. They could come from multiple sources. Putting multiple different purification steps could cost you a lot to set up your manufacturing footprint and break even for your drug and vaccine. So we from a component perspective, we are cautious of how we could support our products innovations to cause less of those impurities after IVT synthesis, for example, double-stranded RNAs, other impurities, right? So these are the steps that could potentially bear some additional cost, and innovating and helping solve for those challenges is definitely worthy.
Thank you, Tadas. I'm being pinged by our organizers to say we're actually out of time, and just remains for me to we could talk, I guess, much of the day on this and beyond, but it's been a real pleasure to pull this panel together, and thanks everyone for your great contributions. I've saved you from the provocative question because we've run out of time, but someone asked, not quite in these words, whether mRNAs were the death of small molecules, but unfortunately we're out of time for you guys to give your answers, so I'll leave the audience to take a look at the facial expressions as we hand back over to Tony. But thanks again, everyone. This has been super.
Thanks, Tim.
Thanks very much, Tim, and indeed to the panel for sharing those insights across such a broad range of challenges. I think it's great that success gives you momentum, as an example. I think we've seen that through many cycles, whether it was years back in immuno-oncology and in cell therapy, you needed examples to get to market and through. And that brought so much attention and investment into the space, which there are challenges to face, both in R&D, but also in and indeed in distribution and access globally, I suspect. But thank you all for sharing those insights, and indeed thanks to Ian for his scene setting and where do we go in the future of R&D thoughts earlier on. So it remains for me just to wind up the session, I guess. Thanks to everybody. We now have a coffee break for 15 minutes. I guess what do you do with that 15 minutes? If you're sat remotely, well, I guess you could check out the on-demand content you might want to go and watch later. You could continue with surfing the delegate list to see who you want to partner with and arrange some more partnering or networking. Or indeed, you could go and grab that coffee and come back and be refreshed, ready for 10:30 when we're looking about how do we harness all of life sciences for better outcomes. But for now, thank you very much, and we'll see you soon.